Alrighty, so we are going to start with chapter 18 now, and we're going to probably finish up chapter 18 and 19 before we start reviewing for our AP exam coming up at the end of May. So our objectives are that we're going to be justifying the selection of geological, physical, and chemical data that reveal Earth's early conditions, describe and evaluate scientific hypotheses about the origin of life on Earth, which is super important and super cool. And then we're also going to evaluate the accuracy and legitimacy of data to answer scientific questions about the origin of life on Earth. So the reason we're doing this is because there are multiple hypotheses about how life started on Earth. And I'm going to have you actually research a little bit and write about one of them, at least that you pick. So before we start with that, I have pulled some definitions for some of these terms for you, but I'm going to ask that you do the other ones on your own just by Googling them. That's fine. Or you can use your textbook. So there's scientific theory, hypothesis. I defined LUCA for you. LUCA is actually an acronym that stands for Last Universal Common Ancestor. And basically, it is the most recent population of organisms that all living organisms currently right now all descended from. So we all have one common ancestor. So like that's talking about you, me, it's talking about fish, it's talking about whales, it's talking about cheetahs. All of us have one common ancestor because life had to originate at some point, and that's LUCA. I'm going to ask that you define biomolecules. Abiotic synthesis is the theory that organic molecules formed before life began, and then they built up to make the first cells. And it could have happened just by chance, right? Just the same way that the universe happened from the Big Bang Theory, right? So basically, abiotic synthesis just means an abiotic or a non-living start to what we know now as like living things. I'm going to have you define what a protocell is. This is kind of interesting to think about. I'm actually going to mention it later, but you have to know the definition of it before I can talk about it. You have to know what a micelle is, a liposome is, and a coacervate droplet. So coacervate droplet is a droplet of liquid that is densely packed with biomolecules like polymers, proteins, or nucleic acids. And this is actually a term used to describe the first protocells that existed. So after you look at the definition of a protocell, this will make a little bit more sense. We're going to also start the chapter 18 Learn Smart and Summary. I will have that assigned in McGraw-Hill for you to complete as we do the notes together across this week and next week, because this week is really, really short, obviously. So what happened from the origin of life is that there was early Earth with nothing. It was technically barren because there was no life forms. And so stage one happens where organic monomers develop. And organic materials basically are anything that contains carbon. And so there are monomers, which are simple carbon-based molecules, developing as molecules collide in the atmosphere, or just by chance, or maybe, for example, when lightning strikes, crazy things happen. In stage two of developing and going towards life on Earth, organic polymers start developing. And that means that these monomers are becoming complex carbon-based molecules. So they're building chains and stacking and coiling, and they're either built or derived from the monomers that started existing a little earlier. Now, stage three is where we get a little bit more interesting. This is where I mentioned protocells that you should have looked up earlier. So protocells are self-organized spherical collections of lipids, fats, that were suggested to be the step towards the origin of life. So they were the prototype of a cell, aka like the rough draft of a cell. And they were circular, and they kind of had that, how we have a phospholipid bilayer as our membrane. They had a lipid membrane, and they had contents on the inside contained by this membrane. So their structure was the like blurry, I don't know, 240 frames per second version of what we currently have as cells. And then stage four, from the protocells, we eventually developed living cells, which are cells that contain a plasma membrane and are considered alive with genetic material that they can pass on. And from cells, you just go on and on and on into more and more complex organisms and structures until we get what we have right now. And it's pretty amazing that this all happened because there's no specific definitive answer to what happened because there's nobody who was alive back then when life first happened, right? So everything is kind of predicted, or we're using evidence to kind of explain, the hypotheses that were possible for how all of these things happened. So the evolution of a protocell is kind of where the first life form came from, right? So why would nutrition be important for the first protocells? Well, protocells need to intake materials from the environment in order to build their polymers and monomers inside. And by doing so, they are able to live, and eventually with enough complexity built into them and growing not organelles, but structures inside of them that help them survive and function better, they eventually evolved into what our first life form cells were. A good question that a lot of people ask is, was oxygen available when the first cells evolved? If not, when and how did oxygen increase in the atmosphere? Now, oxygen only became available 2.33 billion years ago, or BYA. But the first cell actually evolved 3.8 billion years ago. So cells were around before oxygen was around, which is crazy to think of because honestly, like we can't necessarily wrap our heads around this because we physically must use oxygen to survive, right? But the cell actually came first. Oxygen started increasing the atmosphere once cells developed the ability to photosynthesize, right? Because some cells even incorporated others into themselves and became the early ancestors to what we now call as plants. And the ability to photosynthesize takes light energy and helps make oxygen as a byproduct of making glucose. Now, the oxygen was increasing because more and more organisms were able to do photosynthesis or any other form of chemosynthesis that was maybe also producing oxygen. And as oxygen increased, life forms were able to be bigger and more complex because cellular respiration is a lot more efficient when you have oxygen than when you don't. Next question is, what is the difference between chemical evolution and biological evolution? Well, chemical evolution is the eventual formation of more and more stable molecules from smaller forms. So it's essentially the building of the monomers into the polymers into the protocells. But biological evolution is what we normally think about when we talk about evolution. And that is the genetic changes in a population over generations. And that is very, very, very much only applicable to the last couple or so millions of years, right? It's not talking about the initial life forms that formed billions of years ago because we don't really have evidence. And we also couldn't track the genetics back then of what was existing. So what are the major events that happen in each of the 4 stages of the origins of life? Well, obviously, like we said before, stage 1 is organic molecules were produced before cells existed. Stage 2, organic molecules formed polymers, so the complexity increased. Stage 3, polymers became enclosed in membranes. Without the membrane, you don't have cells, or you have to have an encapsulation to have an individual life form that is separate from everything else. So that membrane provides that structure. And then stage 4 is that membrane-bound polymers acquired cellular properties, again, whether by chance or just by usage and or increasing usage. So the functions were changing maybe throughout through mutations or just by chance. And then the ones that were more beneficial were increased throughout the cell or increased in kind of popularity or prominence and how often they appeared in these protocells and cells. So I am going to have you do a little essay assignment. This is just a short paper. You're going to choose one of these hypotheses below in the green that explains each of the 4 stages of the origin of life. I'm going to ask that you look them up and read about them and write an essay summarizing what this hypothesis explains and how it explains how life developed through 4 separate stages. I'm going to ask that you include the scientific evidence that supports this hypothesis because every hypothesis of these 3 has a decent amount of evidence that could be used to explain it, but there are still different hypotheses from each other, okay? So make sure that you're explaining it and summarizing the hypothesis using each of the 4 stages that I mentioned in the previous slide. At the end, everybody is going to include a paragraph at the to discuss these 2 hypotheses, the protein first hypothesis and the RNA first hypothesis. And as you can just basically tell from the name of the hypothesis, it's talking about which one evolved first in life forms. So again, you're going to pick one out of the green, but you're going to write both of the purple. Here's some more definitions. I'm going to go over some more definitions and terms that are important for you to understand as we talk about the origin of life and how we are able to analyze things that lived a long time ago, even though we weren't alive. So fossils, we kind of know as a general idea, right? If you think of a fossil, you usually think of bones and a skeleton, but fossils are any preserved remains or traces of ancient organisms. So they can include bones, shells, feathers, leaves, and they can be an entire organism or just part of one, okay? They're not actually the organism itself. It's just whatever has remained after a long time through preservation inside of rock or inside of silt or inside of clay or ice even. Strata is the term that we use for the sheets or layers of sedimentary rock. And they're the layers that we find on the Earth's crust. And if you actually cut a cross section of the Earth, you can see the different layers and the different colors of them. And as you dig deeper into the Earth, you'll encounter older and older layers that were there and contain maybe samples and fossils from a long time ago. Absolute dating of fossils is a procedure used to determine the precise and exact age of a fossil based on the radioactive minerals that they contain in them. It's usually easier actually not to use the dating process on the fossil itself and use the fossil as a sample, but instead to use the rocks surrounding the fossil or the strata. It's a lot more accurate and easier to do that. Radiometric dating of fossils is kind of related to the last one, is using the natural radioactive decay rates of certain chemical elements like potassium or carbon to find out the age of an ancient material. And it's based on how much of the radioactive material is still inside the sample, because while an organism is alive, they are consistently intaking new radioactive isotopes of carbon, for example, because they're always eating or consuming something or intaking carbon through carbon dioxide. And it just so happens that there's a specific ratio of radioactive carbon in the atmosphere, in the environment, in food. And as you're continually intaking it, you have a consistent amount in your body. However, once you die, you can't intake anymore. But the coolest thing about the radioactive isotopes of potassium or carbon are that they are continually breaking down and they have a half-life and they only exist for a certain period of time before they break down. For example, carbon breaks down into nitrogen and some extra, I think, is it protons? Yeah. So after they break down, they're no longer recognizable as a radioactive isotope. And so the amount of radioactive isotopes in a body or in a preserved remains is decreasing. So you can tell based on the current ratio when you get that fossil or that sample compared to the normal ratio of how much radioactive carbon an organism is supposed to contain, you can compare those and see how long ago or how long it took to get to this point where how much they have is compared to how much they're supposed to have and date them based on that. So it's super cool. And it's just amazing that scientists found out how to do this. And like I kind of mentioned, the half-life of radioisotopes is what they use in order to find out the exact time span of how long ago these organisms lived. So the interval of time required for 1/2 of the atomic nuclei of a radioactive sample to decay is the half-life. So when you have a sample amount and half of it has decayed into another chemical element and is no longer radioactive, that's when you have that time span is marked as the half-life. Relative dating of fossils actually doesn't put a specific number or year on when these fossils existed, but you can kind of get a time span of when they existed and relative to each other, how recently a species existed or compared to another species they existed based on the timeline layer of rock that they belong in that you find their sample in. This is why the Earth's crust is amazing and an amazing tool for us to use for dating fossils, because you can find out just how long ago they existed from how deep in the Earth they are. Stromatolites are technically the earliest fossil evidence of life on Earth. They were originally cyanobacteria, aka like blue-green bacteria, that were the first known organisms to perform photosynthesis and make oxygen. So they are part of the reason why we have oxygen today. And boy, are we thankful for that. There were 5 mass extinctions, and then there were more things that have created and happened afterwards that might be leading to another extinction caused by our activity. So the first one was the Ordovician Silurian extinction, and that happened 440 million years ago, or MYA. And that's when small marine organisms died out. Then the second most recent one we had was the second oldest extinction, was the Devonian extinction, which was 365 million years ago. And that's when a lot of tropical marine species went extinct. And so we see these through fossils that we pick up and dig out. The third one, getting closer to current time, only 250 million years ago, was the Permian Triassic extinction. And this was the largest mass extinction that happened on Earth, and it affected a huge range of species, including a lot of vertebrates or animals that have spinal cords. The second most recent Triassic-Jurassic extinction happened only 210 million years ago, and it was the extinction of other vertebrate species on land. And because these species went extinct, dinosaurs actually became the most common life forms on Earth. And then the most recent extinction that has occurred already is the Cretaceous tertiary extinction, which was only 65 million years ago, which when you compare it to the billions of years that Earth has been around, isn't that long ago? And so non-avian dinosaurs got wiped out. And avian means bird-like. So basically, this is the image that you think about when you think about extinction and the meteor shower and all these dinosaurs dying. But there are a lot of smaller species of dinosaurs and bird-based dinosaurs that survived and are the ancestors to a lot of the things, organisms that we observe nowadays. And there is a prediction that because human activity has wiped out more species in the last 200 years than in the last million years caused before humans, that there's going to be another mass extinction based on human activity very, very soon, as recorded in history, just because we've been wiping out species like no other. So the last page of definitions that we're going to go over are kind of some basic things that we have talked about a little bit in the past. So there are 3 domains of life. The first one is archaea, the second one is bacteria, and the third one is eukarya. So archaea are prokaryotic, no nuclei, extremophile cells. And extremophile just means that they live in crazy environments like volcano vents under the ocean, where there's no sunlight and it's extremely high temperatures. Inside the icebergs of the Antarctic. So inside volcanoes that are exploding and currently boiling lava. So archaea are just like bacteria, except in the fact that they have a different kind of cell wall and that they are only fans of extreme environments. They cannot actually survive in the normal environments that basically all other living things survive in. So if you brought an archaea over to your house and you were just chilling, the archaea would actually die. Bacteria are also prokaryotic, so they don't have a nucleus, but they're commonly pathogenic and they're found in the same types of environments that most life forms that are normal are found. So any environment that we can survive in, pretty much bacteria can also survive in. They might even have a little bit more range on the lower end of the temperature range and the higher end of the temperature range than we do for survival. And they're commonly pathogenic, which means they're infective, but it's not always humans. A lot of bacteria are pathogenic to other organisms because we're not the only organisms that live on this earth. There are plenty of other species for them to infect. Eukarya basically only includes the organisms that have a nucleus. And to be quite honest, the number of species of eukarya is vastly smaller than the number of species of bacteria and archaea. So we are vastly outnumbered by single-celled organisms on the face of the earth. Even in your own body, there are so many bacterial cells inside and on your own body that there's more of those cells than of your own actual human cells. But the size of the cells makes a huge difference. Therefore, you're still by mass, mostly you as a person, but you have a lot of bacterial cells on you and in you. The endosymbiotic theory is the idea that some organelles in eukaryotic cells were once separated and they were once individual prokaryotic microbes. And the evidence for this is pretty self-explanatory because they have their own, mitochondria and chloroplasts are very similar size to prokaryotic cells, and they have their own way of dividing, which is binary fission, very similar to most prokaryotic cells. And they have their own DNA, which is in a circle like bacteria instead of linear like ours. The molecular clock is the average rate at which a species genome accumulates mutations. So basically growing to become different over time. And this is used to measure how often or when they diverge throughout evolution. So speciation can be measured using a molecular clock. And then the last thing is the continental drift theory. And this one's pretty explanatory too. Fossils help to explain it because continents were predicted to have moved around on the Earth's surface. And we all know this idea of Pangea having been this huge supercontinent that existed in the past. And there's pretty good evidence supporting it because, well, there are these fossils of organisms and plants that are found on these random parts of different continents that you're like, well, this organism can't even swim. So how did it travel across half of the earth to then have a fossil found in the tip of a land part of the Antarctic when the other patch of where the fossils are found are in Europe, right? That makes no sense because the animal itself didn't have the capacity to travel that far. So when you show how originally Pangea could have been composed together and you match where the fossil patches are, it's pretty solid evidence for the fact that the continents did move. And we also know currently right now that the Earth's crust is still shifting. And it's partially because we have these things called tectonic plates, and also that nothing really ever is completely still and not in motion. If you take chemistry or physics, you'll actually learn that and about absolute zero. And we know that our oceans, one of them is getting smaller every year by a couple inches, and one of them is getting wider every year by a couple inches. And that's crazy to think about, because maybe at some point in the long, long future ahead of us, the continental shapes will be very, very different. This is the last slide I'm going to share with you. This is the original predicted idea for what Pangea is like, because they have fossils from the Triassic period all the way back throughout history from dinosaurs, where the patches of where the fossils are found don't really match up to our current day continental variation and where they are and location. So when you piece them together based on the fossil evidence, you kind of see this supercontinent that used to exist, and that's where Pangea came from. Go ahead and finish up these notes. Make sure you submit them to get your extra credit today, and make sure that you get started on your chapter 18 summary and learn smart.